Okay, thank you for inviting me to give a lecture in this excellent meeting. My presentation will be a diversion from the clinical topics we have heard and are going to hear later. I would like to present a relatively new general concept of chemotherapy-induced damage to the body, to the organs, and to the patients. And to be more specific, a relatively recent realization that the main effect of chemotherapy is premature aging and senescence caused by the treatment. And due to constraints of time, I will briefly present data showing that there is really premature aging due to chemotherapy. We will discuss several possible biological mechanisms and see how these things pertain to the heart. And so, as you can see, the evidence for senescence by chemotherapy is based on laboratory in vitro data and the clinical data also. In the first slide, the first slide we can see cell cultures, cell lines of different tumors. In this case, these are breast cancer mainly, and they are exposed to radiation or adriamycin. All of them eventually cause senescence of the cells. There are several typical proteins that are elevated and upper expressed during the senescence process. The typical of them are, for example, P16, IMCO4A, and ARF, which represent DNA damage during senescent process. And we can see that in patients treated with chemotherapy, there is a significant elevation of these proteins showing the senescence process is going on. And if we are comparing normal population to people who were treated with chemotherapy, you can see that the senescence markers are much, much higher in patients treated with chemotherapy. Another way to assess premature aging is with clinical data. The aging process is calculated by frailty score, and you can see that there is only one thing lacking in this graph, and this is the control group. But in this population, there should be no frailty score at all. On the other hand, you can see that in all those patients, just a second, in all the patients I am showing here, there are at least 1, 2, or 3 components of frailty score defining them as aging patient, and this is not totally not proportionate to their age, which means that survivors of chemotherapy do manifest clinically and laboratory premature aging. A recent paper published in JNCI summarized the known data about chemotherapy, and you can see that based on molecular markers like P16 and so on, and other clinical and physiological markers, we can summarize that chemotherapy causes an increase in patients' molecular age, more or less about 15 years more than normal aging. It was checked, it was evaluated in survivor of breast and other cancers. In breast cancers, it was 3.5 years aging, and the most significant findings were found in adult survivors of childhood cancer, and in those patients, the premature aging was about 10 or more years. The senescence process is intimately connected with DNA damage, and this is a short scheme just to show us the DNA damage activates ATR and ATM pathways, and finally we don't have time to go over the details of this molecular process, but they finally culminate in senescence, and senescence, at least in vitro, is demonstrated pretty easily by overexpression of the before mentioned molecular markers and staining of beta-galactosidase, which is a typical marker of senescence, and we use it routinely in laboratory settings. The next question is, if that's the point, what can be the main mechanisms of aging by chemotherapy, causing an early onset frailty? And there are basically 4 generally known mechanisms causing aging, like free radical damage, DNA damage, telomere shortening, and neuroendocrine immunologic dysfunctions. I will concentrate on telomere shortening, one because this is the main topic of my laboratory, and the other one because it's probably the most important physiological and pathophysiological cause of aging. So just a brief rehearsal. Telomeres are the non-coding ends of chromosomes, and you can see their DNA has a peculiar structure of loop. The role of telomeres is mainly to protect the DNA from damage, and what's important in our regard, telomeres, due to DNA replication problem inherent to DNA replication, our DNA and telomeres shorten with every division of cells. Therefore, as we can see, that's the length of telomere in normal population. These are data from our laboratory, but these are universal data. Our telomere getting shorter while we age. When the telomeres are too short, they cannot protect the DNA integrity, and they cause up and down regulation. It's called telomere position effect of various genes, pushing the cells into senescence. And that's what happens when the telomeres are shortened. In other words, when the cells or the people become aged. Clinically, telomere length is really connected and related to longevity and survival. You can see data from people with different length of telomeres, and we can see that shorter telomeres are associated with higher mortality, and that's because of aging. 2 examples, more illustrative examples of importance of telomeres in aging and premature aging actually are shown here. This is Dolly, the first cloned sheep. Actually, it's not cloned, it was nuclear transfer, but these are details. And as you remember, Dolly, if you don't, maybe Dolly died unexpectedly 3 years after her cloning. And she died of premature aging. At that time, nobody understood what happened, and it turned out that when they cloned her, they did not take into account the telomere length. This is the telomere length, which, we can see that they started with much shorter telomeres, and that's why they got shortened more and more, and Dolly became an elderly sheep within 3 years and died of old age. Another example of abnormality of aging because of shortened telomere are the classical progerias. This is a picture of child with childhood progeria. You can see this is a 6-year-old child. It has telomeropathy. His telomeres are very, very short, and unfortunately, he will die of old age before age of 10. So, and when we are looking in vitro, we can see that if we, for example, culture human mesenchymal stem cells, we can see the shortening of telomeres and aging of those cells, which means that telomere shortening is the main and principal avenue of aging and premature aging. What happens with telomeres during chemotherapy? We know from multiple studies, many of them originating from our laboratory, that chemotherapy causes telomere shortening in the body. You can see, for example, for every age, 40, 60, 80, that the telomeres of these patients are much, much shorter than of normal population. And the degree of shortening is relative to intensity of chemotherapy. So compared to expected length of 100%, standard chemotherapy decreases the telomere length by 20% and more intensive therapy by almost 30 to 40%. And if we look at the slope of telomeres during the life of patients, we can see, you see the red line is chemotherapy, and they aged or they are shortened much, much more, much faster. Actually, we calculated that it's equivalent to about 15 years of life. Because of time constraints, I cannot show you more data, but we and other groups show that practically all cytotoxic agents, the new tyrosine kinases, and other targeted therapies result in telomere shortening. So we have a universal response, a universal effect of chemotherapy. And is it true for cardiac cells? So we can see that culturing cardiac cells, exposing them to adriamycin, there is immediate upstaining of beta-galactosidase, which means that they really undergo senescence, which means that it's really true for the heart. The same about cardiac progenitor cells. Even they reveal senescence markers after exposure to adriamycin. Therefore, we know that telomere shortening and resulting telomere dysfunction will cause cardiac aging through different mechanisms, but they are one of the causes, and we think that we may regard cardiotoxicity of chemotherapy as part of the general response of premature aging. There are also clinical data showing that shortened telomeres are associated with increased mortality from coronary disease, for example, in elderly people. And the telomere dysfunction, again, is associated with p53 abnormalities in cardiac cells. Is it possible to salvage those cardiac cells? This is a nice example just showing you the possible solutions to this problem, and there are several. The progenitor cells do not shorten the telomeres similar to other cells because they have telomerase, which is an enzyme that enlarges and then lengthens the telomeres. And we really can see that doxorubicin, adria-induced cardiomyopathy, can be salvaged by upregulating the cardiac precursor cells. So that's an option. Another option is we have several emerging methods of attenuating the telomere shortening, and we can see one example. If we transduce telomerase on senescence cells, which is the upper panel, those cells became younger, they start proliferate, and we can, at least on cellular basis, reverse the senescence process. So this is a beginning. A clinical example that I wanted to show you is an experiment we did in our lab and published relatively many years ago. This is the shortening of telomeres after chemotherapy, but we showed that injection of colony-stimulating factor to those patients ameliorates the telomere shortening, and you see the several arrows showing that the telomeres did not get shorter, but actually were lengthened after treatment. So there are theoretical possibilities to, after recognizing the phenomenon, to do something about it, and maybe to intervene. To summarize, the hypothesis is we know that chemotherapy induced telomere shortening, and the telomere shortening is probably the main cause of premature accelerated aging. Chemotherapy-associated cardiomyopathy is a specific example of accelerated aging. That's how would I view it in the broader context. Prevention of telomere shortening attenuates aging, and the question is, what are the future prospects for cardiac damage prevention? Maybe attenuation of telomere shortening during chemotherapy. Thank you.